Yellow fever is a viral hemorrhagic disease that has been around for a long time. It's been in human populations for probably thousands of years, and in the last few hundred years has caused a lot of concern. Yellow fever is caused by a virus. Viruses are interesting infectious entities, primarily because they are not considered to be alive. Viruses, in their simplest aspect, you could consider a virus to be a protein box. It's got a protein shell, and inside that shell is the genetic material that contains the instructions on how to make that virus. This genetic material can either be DNA or it can be RNA. So viruses cause infections by the way they have to replicate. A virus attaches to a cell. It then injects its genetic material into that cell. Once that genetic material is inside the cell, it takes over the cell's machinery. This causes the cell to lose its capacity to perform its normal functions. Instead, the virus has commandeered the cell's machinery to produce the different pieces of the virus. These pieces are then assembled into intact virus particles, and these virus particles are released. Once they are released, they can circulate in the body, attach to uninfected cells, and initiate this replicative cycle again. Viruses, when they replicate inside a cell, can produce literally thousands of copies of themselves. This is why viral infections can be so severe. For yellow fever caused by a virus, the virus contains not DNA, which is most common, but RNA. RNA is an interesting genetic material when it's found in viruses because it has the capacity to mutate at a higher rate than DNA when that is inside the virus. This creates some issues, including more possible variants of the virus. Fortunately, the RNA in the yellow fever virus, unlike other viruses such as the virus that causes AIDS or Ebola, is fairly stable, and there haven't been too many genetic mutants. As we'll talk about later, this allows us then to make a pretty effective vaccine. The other thing that's important to remember about any kind of pathogen or disease-causing organism is they have to have a reservoir or a place where they can hang out when they're not infecting humans. The yellow fever virus has such a reservoir. This reservoir are primates, monkeys that inhabit usually the canopy area of the jungle. These monkeys carry the yellow fever virus. It can cause infection in these monkeys, but this is the place where the mosquito, which transmits the yellow fever virus from the infected monkey to a human, picks up the virus. This makes it incredibly difficult to control this virus because you have to control the reservoir, and that is not practical right now. This state of the virus is called sylvatic or jungle yellow fever, meaning that the virus hangs out inside these monkeys, and periodically when a mosquito bites the monkey and it picks up a drop of the infected blood, it can bite a human in that jungle area, transmitting the virus from the monkey to the human. Initially, when yellow fever scientists began to elucidate how yellow fever worked and that it was caused by a virus, there was a feeling when a vaccine was developed that yellow fever would be able to be eradicated as a health issue for humans. But because the reservoir is common in many jungle areas, particularly in Africa and in Central and South America, scientists rapidly realized this would not be possible. The Spanish-American War is what led to the discovery of the virus that causes yellow fever. As American troops went down to Cuba, yellow fever was a huge health concern. And the Rockefeller Foundation, which had a history of funding health grants to look at significant health problems, commissioned Walter Reed, then a physician in the Army, to examine what was causing yellow fever, what could be done about it. And it was the Walter Reed Commission that discovered a couple of interesting things. This was the first time that anybody had shown that an actual disease agent was transmitted by a mosquito. So that was a significant finding as a result of this commission. And number two, they thought it was probably caused by a bacteria. But when they did some filtering experiments, which means they took the infectious agent and passed it through very small pores in a filter, they discovered that it was much smaller than a bacteria. And at the time, people were thinking they could make a bacterial vaccine to prevent yellow fever. They discovered instead that it was caused by an infectious agent much smaller than that, and they soon realized they were dealing with the virus. So yellow fever has a unique place in the history of human disease, being the first viral disease, human viral disease, to be isolated, the first human viral disease transmitted by mosquitoes to have that discovered. And as a result of that, opportunities then became available to make a vaccine.